I want to put a little bit a different spin on bioinformatics in the biotech industry. I've been working there for quite some time, and we've encountered a couple of things which I would have never thought I would encounter when I left the academia. But first, oops, what do I have to do? Yes, I'm terribly sorry about that. Yay. But first, who about you has ever heard of DSM or DSM nutritional products? I did, and a couple of others, 20% of the audience. That's not too bad. The thing is, we are one of the world's leading suppliers of vitamins, carotenoids, nutritional lipids, and things like these. But not too many people know us. That's because although we have a business of something like 4 billion euro, we sell only to industries and not to consumers. We sell to feed, to food, to pharma, and personal care. And basically, the majority of you will not get through the day without eating something where we won't have supplied the ingredients, either directly for your food or for the feed of the animals you eat. If you think of that, the products we are selling are either extracted from nature, they are produced by chemistry, and that's the reason why I'm here, they are produced by biotechnology, and biotechnology means bioinformatics. Yay. Two disclaimers first, DSM nutritional products, we don't sell anything regarding bioinformatics products. We don't lease that, we don't rent that. I don't want to sell you anything. Just eat your vitamins. Please eat fruits or vegetables. That's all I want to say. However, as we are not working in this field, or we are not interested in bioinformatics as a product, we are also allowed and strongly encouraged by management to work with public projects and open source software, not only using it, but also contributing it or having even own projects. The second disclaimer I'm making is our industry is producing, our company is producing low price products. If we were to sell a kilogram of vitamin C for $1,000, we would be very happy, but we would have no customers. That's selling for a price point below $10. So what I'm going to show you may be applicable to you, but not necessarily so. Let's start small, because if you think of computation, well, you think that you may need a lot of computing power. You don't. A typical thing, you have a favorite production organism and you want to produce a product called XYZ. It doesn't do it by itself, but fortunately in literature there is, for example, a publication describing a gene XYZA, which does what you want, and you want to put it into your organism. There's also RNA-seq data of that organism. So a typical question at one point in time from your biologists would be, for example, to have a check on the sequence as was published in the literature against what is really there. If you're a bioinformatician, your answer could be, yes, it's 99.8% identical. That's a very good answer, but if you're working in a team, you should think a little bit more. You should look a little bit on the sideways. For example, there's also a similar relative of that gene, 70% similar, less frequently expressed, has no name, not described anywhere. And the nice thing about working in industry, and if you have a nice group of colleagues who are really, really enthusiastic, they say, okay, we won't try only XYZA, but we will also try the similar gene. And in that case, if XYZA, the gene itself, produced 100% of the product you wanted to have, well, by chance, the similar gene got you immediately 300% of that product. And that's quite important, because if you think of it, 3 minutes of your time saved several $100,000 of development cost, because we already had plans for trying out different promoters, for trying out multiple gene copies. If that wouldn't have helped, we would have gone into protein engineering. No, all that completely fell flat. And BLAST, I'm sorry, that's something your laptop can do. You don't need a huge cluster for that. Let's get a little bit slightly larger. I'll do a small detour for that. There is a very nice paper from the beginning of this year which is called How Many Biological Replicates Are Needed in an RNA-seq Experiment. I encourage you to go out and read it. It's a very good paper, and this is a spoiler alert because this is the gist of it. The recommendations for RNA-seq experimental design are you should have at least 6 biological replicates, and if you can have that, well, maybe 12 of them would be better. Now, let me show you something. This is a real image of one of our production sites. If you look at this structure down here, and as this is Google Map and Google Earth, you can zoom down to that, and you can rotate it, you get this. And if you have a little bit of an imagination, you can see this is a train. Oh, well, at least a locomotive and a wagon. The wagon is the most interesting thing because this is the raw materials for one fermentation. And honestly, I'm not even sure that it's enough for one of our fermentations. And if you now start to think of, okay, if that is what you need for do one fermentation, and this is at production scale, how difficult is it to have 12 or even 6 exact replicates? Because you have different batches of your raw materials, be it glucose, fructose, or any other things. You don't get exact batches. Then you have problems, for example, that you can't always use the exact same fermenter, or you don't always have the same personnel on site, etc., etc., etc. So basically, often you have the problem that you can't have exact replicates, and we do RNA-seq or chip expression experiments on this kind of scale because their organisms behave a little bit differently than in shake flasks. Now, if we have new technologies every couple of years, we want to use them because hopefully they will help us to get better, faster, whatever not. So we want to know if there's a new technology, how can we use it? Just a small example, this is from when Solexa Illumina started to be interesting. Let's say you have a production organism which has 150 differences to the wild type organism. And then you might want to know, okay, what are deleterious mutations, what are good mutations? So you have a project, it's a little bit a gene-wide association study in the light version to get a feeling of what mutations can I get out of this organism. So the chosen approach is you put in the mutations into the into a wild type strain, and you do that for a number of strains. Let's say that there was one of those strains which was not really behaving as expected. What do? Well, first your biologist will tell you, well, this is our strain, it's a bacillus. We engineered those 3 mutations into that strain. We worked quite well because we did PCR, we know exactly what we entered. At that time, we said, okay, Solexa is a new interesting technology. Let's have a look and see what we can see with that. Maybe there's some silly mutation having entered the strain. This is a coverage plot of Solexa. Even if you have never seen that, you will quickly see that this here is not normal. This is a 200 kilobase duplication. Whenever we showed that image to biologists, they were telling us this is almost not possible that this is stable. It should loop out within seconds. No, it doesn't. And this is showing you, okay, technology can be used for interesting things, also for quality checks. So if you think of that, you think, okay, maybe we want to use that a little bit later, also automatically or automated in an automated way, because we are never sure that something silly doesn't happen in the organism when you engineer it. So this is from 2016, actually from May. This is again a coverage plot of an organism. And if the black line here is the average coverage, the red line here is twice the average coverage, then every self-respecting peak finding algorithm will tell you that this is a 2 kilobase duplication. Don't even think of questioning it because all the p-values or certainty values that you can calculate will tell you that's a sure thing. When I looked where the second copy of that thing were to be integrated into the genome, I didn't find anything. And then I suspected something I've seen already in the past, and this is here a GC plot of the organism. And if you know that the organism has an average GC content of 57%, this special area has 32%, you see Illumina is somehow lying to you. The thing is, Illumina does not always lie to you. We've seen that earlier. This is data from Solexa still, 2008. This was one of the very first Solexa data sets that we had. It's a very nice coverage plot. We like that. The very same organism a couple of months later and up until 2010 looked like this. The very same organism. And if you think that you're running an automated peak finder to find mutations there, you will find approximately 577 mutations. No, it's not the case. This is simply data noise. This is something most people don't know that can happen because it's not too much described in literature. Even the sequencing provider often do not know about that. Let's shift gears. We are bioinformaticians, or at least some of us, and we build something which is called models because, well, we try to explain the world. And once we've got those models, we try to predict things which have not been seen before. Things not always go smoothly. So this is a pretty simple model. Glucose enters the cell, goes down to the TCA cycle. This is a problem we had in 2004, 2005. We are not interested really in that. There is a side pathway, which is called the pentose phosphate shunt. We are still not interested in that. We are more interested in riboflavin, because riboflavin, being vitamin B2, we want to produce it, lots of it. So you have a project to increase the production of furin biosynthesis and riboflavin. And if you let run your algorithms, for example, to do flux analysis and carbon balance, you might get an answer that to increase riboflavin, well, if you knocked out or attenuated TKT, the gene which is the second entrance to the pathway, you might get what you want. So that's quickly done. Your biologists create a delta TKT mutant. And the first thing they will notice after that is, well, that organism doesn't grow on glucose anymore. We just deleted a gene and it doesn't grow on glucose. That was a little bit unexpected. Second thing, there are some colonies spontaneously recovering from that. So there is a mutation which helps the organism to recover the growth on glucose. At that time in 2004, our people tried to do a wet lab mapping because sequencing was not really there. They didn't find the mutation. Fortunately, 2005, a company called Nimblegen offered something which was sequencing by, I even forgot about that. Anyway, it's not, it's history now. Everyone uses Illumina. So they told us they found 4 or 5 mutations, and one of those mutations was in VWF, the other entrance to the pathway. So at that time, everyone, including me, got a little bit excited because, yes, that's probably increasing the flux. It's hyperactive protein. It's a logical and very sexy thing to think. So you sequence, at that time still by PCR, 19 other independent colonies, and in 16 of them, you find an amino acid mutation in that gene ZWF. Good. We know everything we want to know. That's the gene which is interesting to us. Do we know what those things are doing? If we wanted to do that just by amino acid mutations, you would need to perform a little bit of computation. Fortunately, those were not the only mutations. We sequenced 20 of them. Those are 70 of those mutations. The 3 other mutations were those stop codons. And at that time, you have to come back to the pathway. That pathway had 2 entrances. We nuked out the first one. And the best thing this organism found to recover growth on glucose was to nuke out the other one, which is also an entrance to the purine biosynthesis pathway. It should be dead. No, it wasn't. You probably, basically, also our biologists constructed the delta TKT, delta ZWF mutant by themselves, just to be sure. Yes, it happily grows on glucose. You probably now can imagine what happened at that point in time. There was a 3rd entrance to that pathway, which appeared a couple of months after that in the databases. But this is a pathway which even in 2004, 2005 had been under scrutiny for tens, if not dozens of years. People thought we know everything there is to know about that pathway. Unfortunately, no. So, basically, we still continued work on TKT, and a couple of years later, we got what we wanted. Yeah, I'm basically almost ready. What we could have done working on that in high throughput mode at that time would be look at chemical properties, conservation analysis, all those kind of things. Nowadays, medium throughput, you might want to analyze also automatically by text, nearby annotated regions of interest. Low throughut, still low throughput, if you compare that to other methods nowadays, would be 3D modeling of mutated sites versus regions of interest. But the fact that we found the stop codons meant that we didn't do to have all this work. So basically, if you can avoid bioinformatics and computation just by running a couple of more of experiments, you should do that, because they will more probably tell you the truth much more quickly than what everyone thinks is, than everyone else by computation. So this, animation works. This would have been a model, but I'm not going into that. So the challenges that we have in the bioinformatics industry. I think we are at the forefront of technology and biological knowledge development, not the industry itself, but the whole field. And that means that we have a lot of moving parts. That's a highly complex field. We have new game-changing technologies every few years. If not, then we have new kits or new protocols every few months, and they will change your data ever so slightly. All of them do, or well, most of them. Then there are new good algorithm programs every few weeks, which are published, not the least of in bioinformatics, for example. And then, of course, biologists are not sitting without doing anything too. So there's description of new genes, new functions every single day. And with that, we are building an enormous amount of technological debt, where one of the main problems is to back integrate the knowledge from a couple of years ago into the new models that we have today. That just showed you also the replicability is not always possible. So if n equals 3 or n equals 6 in replicates is not possible, we need to find ways to work with n equal 1. And sometimes we lack a little bit the methods to do that. And there's one of my main pet peeves, and that's the hubris of bioinformaticians. We think that our models and our organisms represent the reality. And because we are putting so much work and so much computing power into that, we think that the results must be correct. And actually, no. Most of the time, nowadays still, we have probably more than a 50% chance of giving the correct answer, but it's not 100%. And we should always be on the lookout for things which we did not expect. And with that, that was a last slide, which was to reinforce that last idea. But as we don't have time, thank you for your attention.